Hello everyone, welcome back to my channel. Today we'll discuss the histology of cartilage. As we know, cartilage is divided into hyaline, elastic and white fibrocartilage. The hyaline cartilage is the most abundant cartilage in our body and it is seen in the fetal cartilage, the tracheal rings and few cartilages of the larynx, the articular cartilage in many places. It is covered by a connective tissue sheath called perichondrium. And cartilages are avascular, so no need to write the blood vessels. The outer fibrous layer and inner cellular layer which contains the chondroblast which multiply and give rise to chondrocytes. Coming to the main matrix, it is glassy appearance, that's why the name hyaline. The hyalose means glassy. And it is rich in sulfated glycoproteins produced by the chondrocytes. It also contains a lacunae. Lacunae are the spaces. Inside those spaces, we can see the chondrocytes. Chondrocytes are little bigger in size compared to the chondroblast, and they are always present in groups. That is called nest. The one chondrocyte will multiply and give rise to the daughter cells, and they are present in the groups. They will be present in group of 4 to 8. And each cell is surrounded by a lacunae, the space, and they have a central round nucleus. The matrix is 60 to 80% water and 2 to 3% of cells. As it is rich in water, when the cells secrete sulfated glycoprotein, it diffuses into the watery matrix. So imagine a paint dropping into the water, it diffuses, no? Where it is produced or where it is produced, it's more darker, which stain blue. That's a hematoxylin stain because its sulfur content is more. So you have a densely stained matrix immediately surrounding the cell. That is called capsular matrix. So the surrounding a cell nest, the density is little lesser, but it is compared to the interterritorial matrix, it is more dense. So that is a territorial matrix. It's a density of sulfated glycoprotein surrounding the nest. Interterritorial matrix is between the cell nest. The density of this sulfated glycoprotein is very less. So that's why it is little lighter staining. It's a concentration of sulfated glycoprotein which causes the gradation in the staining of hematoxylin. Here we are not going, matrix is full of collagen fiber type 2, but we are not going to write collagen fiber here because the matrix, the refractive index of the matrix and the collagen fiber is the same. We won't see any fibrous tissue in the matrix. Here I am showing a separate thing where the capsular matrix, which is a highly dense hematoxylin stain, and a cell nest showing a territorial matrix surrounding in the separate slide. So make sure capsular is immediately surrounding the cell, territorial is the density surrounding the nest. See the density, the color shading. Coming to the elastic cartilage. The elastic cartilage is present wherever you need a flexibility. It is present in the ear pinna, auditory tube, and also in some of the cartilages of the larynx, epiglottis and all. So similar, this also has a perichondrium which has outer fibrous and inner cellular layer. Here the chondrocytes are present in the matrix, either single or in a group of 2. Sometimes 3 cells also can be present. These cells are present in a lacunae, the space. They are little bigger than in compared to hyaline cartilage. Make sure to write a lacunae, the central round nucleus, and the surrounding matrix is filled with highly branching and anastomosing thin elastic fibers. You also find type 2 collagen fibers. but it is very difficult to distinguish between the elastic and collagen fibers in H&E stains. So make sure you write the branching and anastomosing elastic fibers which completely fill the matrix surrounding the cells. This is elastic cartilage. Coming to the white fibrocartilage, very important thing is it doesn't have perichondrium. It is not surrounded by perichondrium and it is very rich in dense regular connective tissue. It is like a transition between the dense regular connective tissue and with the cartilage. The only difference is you can see the cells which are arranged in rows in the white fibrocartilage. This is called white because lot of collagen fibers gives a whitish appearance. Here the bundles of regular connective tissue and the cells will be present in the lacunae in a rows amidst the collagen fibers. No perichondrium. That's the main difference here. The cells are similar to the chondrocytes are similar to the other cartilages. And it is seen in intervertebral disc, symphysis pubis and in the labrum, glenoidal labrum. I hope these diagrams are helpful. Please like, share and subscribe to my channel. Thank you.